In the early spring, two salamanders begin their courtship and mate. Then, responding to an ancient instinct, the female folds a leaf into an underwater cradle, where she lays a fertilized egg. As salamander eggs grow into embryos, their early development parallels the growth of human embryos. So we can learn more about ourselves by collecting amphibian eggs and watching them grow in the laboratory. The eggs will reveal a process common to all animals on Earth, as one-celled eggs become complex individuals with billions of cells, each cell specialized to perform a specific function. The cell divides into two cells, the two into four. The embryo is developing. As cell division goes on, each of the cells looks much the same at first. Inside the developing embryo, an open space is forming, creating a hollow ball, or blastula. As cell division continues, a dimple called the blastopore begins to form as cells start migrating inward to line a newly developing cavity. As cells move in, some are destined to become embryonic muscle, some stomach and intestines. A last movement of streaming cells pushes in nearly all the yolk-filled cells. Eventually, only a small plug of yolk cells remains on the surface of the developing embryo. This yolk plug area will become the embryo's posterior. The other end will be its head. If we look at the embryo from above, we will see its nervous system begin as two parallel ridges of tissue along its back. Gradually, the ridges fold over and fuse together, enclosing cells that will form the brain and spinal cord. Originally, the cells simply increased in number. Now, they're also becoming specialized or differentiated, forming specific tissues and organs. Scientists have devised many experiments to study differentiation more closely. To embryologist Hans Spemann, the embryo was, in his words, a conversational partner who must be permitted to answer in his own language. Spemann's genius lay in asking the right questions. One question he asked was, when do permanent differences appear among an embryo's cells? He wondered, does it happen sometime during cell division? Or is it determined at the very beginning in the substance of the undivided egg? If that is so, is one half of the egg committed to building a head, gills, eyes, a brain, and the other to building only the rest of the salamander? Speman found a unique way to determine if the halves of the eggs were already committed to these separate directions of development. First, he tied a strand of baby hair into a loop. Then, just before a salamander egg divided for the first time, he slipped the loop around it, dividing it vertically. Spemann reasoned that if the two halves of an egg are already differentiated, then one half might form a head, while the other might form a tail. In fact, something different happened. By the end of the first day, both halves of the egg were growing separately, except where they shared a few cells within the loop of hair. Each side had developed its own blastopore and underwent cell migration. Soon, the embryo developed two complete spinal cords. When the experiment was performed with the loop tied tightly, two separate, complete embryos formed. When the hair was tied loosely, a secondary embryo formed that was connected at the location of Spemann's loop. If each of the halves can form an embryo, then permanent differences must not begin until after an egg has divided, perhaps several times. Spemann tried dividing the embryo after it had developed a blastopore, so that the blastopore was only in the dorsal half, on the left here. He found that this half will form a spinal cord. It will continue to grow, almost normally, differentiating into complex organs and tissues. But the other half, without a blastopore, will remain a stunted, unorganized mass that Spemann called a belly. Spemann concluded that the formation of a blastopore marks the moment when permanent differences start to appear among the embryo cells. Is it possible to determine the future of the cells that migrate through the blastopore? Another embryologist, Walter Vogt, by staining parts of the developing embryo with harmless dyes, was actually able to follow the migrating cells. With a good microscope and a steady hand, we can repeat Vogt's experiments. First, we'll prepare small bits of colored agar to act as tiny paintbrushes. We place these bits of agar on the surface of an egg, one by one. Now the bits are removed, but the stains remain. Dyed cells migrate normally into the blastopore, unaffected by their tags of color. The cells seem especially active on the upper or dorsal lip of the blastopore. A cross-section of the blastula would show its hollow interior, the blastocele. Above is a single layer of tissue, called ectoderm. Tissue below is called endoderm, formed of large yolk-filled cells. Here on the lower right are some of our initial colored stains, about to move into the blastopore. Stained cells continue to migrate through the blastopore. Inside, some cells are forming mesoderm tissue that lines the roof of a new cavity called the archenteron. This cavity will become the digestive tract. Now we add new areas of stain to the outer surface of the developing embryo, the ectoderm. As the yolk plug disappears, two ridges form along a thickened area of cells on the embryo's back. The ridges fold over and join, encasing the thickened area that will become nervous tissue. Over it, a thin, transparent layer of other ectodermal cells will form the embryo's skin. Underneath, dyed cells are still faintly visible. They are elongating, pushing their way forward inside the embryo's body to form the spinal cord and brain. On the basis of such observations, Walter Vogt constructed what he called a fate map. It outlined several areas of cells on a blastula's surface and predicted their future development. Folks suggested that adult organs can, in every case, be traced to specific regions on an early embryo's surface. We can check this idea by watching the process backwards. Spemann began to suspect that one part of an embryo might induce other parts to develop in certain ways. Spemann and his assistant, Hilda Mangold, asked the question, what would happen if cells from one region of an embryo during its early development were transplanted to another? They found that when cells destined, for instance, to become nervous tissue were transplanted into a region of future epidermis, the cells became epidermal cells. But what would happen if the dorsal lip of a blastopore were cut out and moved? To recreate Spemann and Mangold's experiment, we can prepare a salamander embryo that has just formed a blastopore. To prepare the cell for microsurgery, we need to peel away a thin membrane. This will allow us to get at the embryo's tender surface. In another dish, we have a second embryo ready. This one has been dyed blue. The membrane must also be removed from this cell. When a bit of its tissue is transplanted to the first embryo, the blue color will indicate the grafted cells. With a fine tungsten needle, we cut out a small group of cells from the blue embryo's dorsal lip. This will be our graft. On the undyed embryo, the graft's host, we cut out a matching group of future epidermal cells that are not close to its blastopore. Now the extradorsal lip, stained blue, is slipped into place on its host surface. We can observe that the host embryo now has an extra dorsal lip. As the embryo recovers from microsurgery, cell migration begins at two different sites. Cells migrate inward through the embryo's blastopore and through the new blastopore that has formed under the grafted dorsal lip. Now there are two centers of organization. After 3 days' growth, the embryo begins to form two spinal cords. Our embryo is becoming two organisms joined together. From this and other experiments, Spemann determined that the grafted dorsal lip induces cells around it to change their fate. Different structures are induced depending on where the transplant is made and what part of the dorsal lip is transplanted. Two heads may be formed, or two tails, or two hearts can form, beating in delicate syncopation. At the dorsal lip of the blastopore, Spemann had found the organizer, that group of cells that differentiates first and induces the differentiation of other cells around it. In part for this achievement, Spemann won a Nobel Prize in 1935. His and Folt's brilliant experiments opened a whole new field of investigation into one of the central problems of biology, how the cells of a fertilized egg are guided in fulfilling their differentiated destinies.